There is a fruit sitting in most people's refrigerators right now that has been tested directly against cancer cells in multiple independent laboratories. And the numbers coming out of those studies are extraordinary. Colon cancer cell growth inhibited by more than 50%. Tumor incidence in breast cancer models reduced from 96% down to 61%. Tumor volume reduced by 45%. Natural killer cell counts, the immune cells that hunt and destroy cancer, significantly increased after 6 weeks of daily consumption. Inflammatory adhesion molecules on blood vessel walls suppressed by 86 to 98%. These are not vague claims. These are specific numbers from peer-reviewed studies published in scientific journals. And the food producing these results costs about $4 a pint at any grocery store. That food is the blueberry. Now, I want to be honest with you right from the beginning. As a physician who studies this research carefully, I know that laboratory results and human clinical outcomes are not always the same thing. I am going to tell you what the laboratory science shows. I am going to tell you what the human studies show. And I am going to be clear about the distinction. Because on this channel, we believe that honest science education, not exaggeration, not hype, is the only kind worth doing. But here is what I can tell you with confidence. The science on blueberries is some of the most consistent, most replicated, most mechanistically understood nutritional science in existence. And by the time this video is finished, you are going to understand exactly why Dr. William Li identifies blueberries as one of the most important foods you can eat after 50. Stay with me, because the numbers I am about to share with you are going to change how you think about this small blue fruit entirely. Let us start at the beginning. What makes blueberries different from other fruits? The answer is a compound called anthocyanins, the pigments that give blueberries their distinctive deep blue-purple color. Anthocyanins belong to a broader class of plant compounds called flavonoids, and they are one of the most extensively studied categories of natural bioactive substances in nutritional science. Here is the first number worth knowing. In a comprehensive analysis conducted at Tufts University that examined 40 different fruits and vegetables for their anthocyanin content, blueberries ranked highest of all 40. Not highest among berries, highest among every fruit and vegetable in the study. That exceptional concentration of anthocyanins is the foundation of everything else we are going to discuss today. And within blueberries, there is an important distinction that most people do not know. Wild blueberries, the small, intensely pigmented variety native to North America, typically found in the frozen section of grocery stores, contain significantly higher anthocyanin concentrations than the larger cultivated blueberries most people buy fresh. This matters practically, and I will come back to it at the end of the video. Now let us look at what those anthocyanins actually do inside the human body. We are going to go through this system by system, using the framework of Dr. William Li's 5 health defense systems. Defense System 1? Immunity and natural killer cells. Dr. William Li specifically identifies blueberries as a food that boosts natural killer cells. These are the specialized immune cells that patrol your body looking for abnormal cells, including early-stage cancer cells, and destroy them before they can develop into tumors. We covered them in detail in the immunity video, but today I want to give you the specific numbers from the research. A study published in Applied Physiology, Nutrition, and Metabolism examined the effect of blueberry consumption on natural killer cell counts in human subjects under the physiological stress of prolonged exercise. Here is the protocol. Subjects consumed 250 grams of blueberries per day for 6 weeks. On the day of testing, they consumed an additional 375 grams 1 hour before running for 2.5 hours at approximately 72% of their maximum oxygen consumption. Blood samples were analyzed before and after exercise for natural killer cell counts, inflammatory markers, oxidative stress, and other immune parameters. The result? Blueberry ingestion significantly improved natural killer cell counts both before and after the exercise bout, while simultaneously reducing markers of oxidative stress and inflammation compared to the control group. Now, I want to explain why this matters specifically for adults over 50 who are exercising, which should be all of you, because the evidence for physical activity and long-term health is overwhelming. Here is the phenomenon you need to understand. Strenuous or prolonged exercise creates a temporary state of immune suppression. In the hours immediately following intense physical activity, natural killer cell activity measurably drops. Your body is redirecting resources toward muscle repair, inflammation management, and metabolic recovery. And during that window, sometimes called the open window theory in exercise immunology, Your immune surveillance system is running at reduced capacity. Your ability to detect and destroy abnormal cells is temporarily diminished. This is not a reason to stop exercising. The long-term immune and cancer protective benefits of regular physical activity massively outweigh this short-term effect. But it is a reason to think strategically about nutrition around your workouts. Because what the Blueberry study demonstrates is this: consuming blueberries before and after exercise can close that immune window. The anthocyanins appear to maintain natural killer cell activity even during the recovery period when it would otherwise decline. For adults over 50, whose baseline natural killer cell activity is already naturally lower than in younger adults, this protective effect may be especially meaningful. The practical protocol from the research: a handful of blueberries, roughly 80 to 100 grams, about a cup, before your walk, your swim, your yoga session, or your workout. Another handful after. That is it. A separate study published in the journal Molecules examined the effect of a specific blueberry polysaccharide on tumor-bearing mice. The results were striking. Tumor growth rate was inhibited by 73.4% in the treatment group. NK cell activity improved significantly compared to controls, with p-values below 0.01, indicating a high level of statistical confidence in the result. The blueberry compound also increased spleen and thymus indices, measures of immune organ health, and boosted the production of key immune signaling molecules, including TNF-alpha, IFN-gamma, and IL-2. These are the molecular signals your immune system uses to coordinate its cancer-fighting response. Blueberry compounds increased all of them simultaneously. Defense System 2, angiogenesis. We have discussed angiogenesis extensively on this channel. The ability of tumors to recruit new blood vessels, to build their own private blood supply, is what transforms a microscopic, harmless cancer cluster into a dangerous, growing tumor. Your body's angiogenesis defense system works to prevent that recruitment, and certain foods have been shown to support that defense. Blueberry anthocyanins have demonstrated anti-angiogenic activity in laboratory studies. Specifically, the malvidin glycoside anthocyanins found in blueberries, malvidin 3-glucoside and malvidin 3-galactoside, have been shown to inhibit the expression of inflammatory adhesion molecules on endothelial cells, which are the cells lining your blood vessels. These adhesion molecules, including ICAM-1 and VCAM-1, play a role in the inflammatory signaling that supports abnormal blood vessel growth. Here are the specific numbers. At a concentration of 10 micromoles, the mixture of these two blueberry anthocyanins inhibited increased MCP1 production by 86.8%, ICAM 1 by 98%, and VCAM 1 by 70.1%. These are not modest reductions. These are near-complete suppressions of the inflammatory signals that contribute to pathological blood vessel growth. Now, laboratory concentrations do not always translate directly to the concentrations achievable through normal food consumption. But these numbers tell us that the mechanism is real, that blueberry anthocyanins are biologically active against these specific molecular targets, and that the direction of the effect is consistent with what we would want for cancer prevention. Defense System 3, DNA protection. This is where the numbers get particularly compelling. Multiple independent research groups have now tested blueberry anthocyanins directly against cancer cell lines in laboratory settings. Study 1, colon cancer cells. Researchers tested blueberry phenolic compounds against two established colon cancer cell lines, HT29 and CACO2. The anthocyanin fraction of blueberries inhibited the growth of both cell lines by more than 50%. The same anthocyanin fractions also resulted in 2 to 7 times increases in DNA fragmentation in the cancer cells, a direct measure of apoptosis, meaning the programmed cell death that eliminates abnormal cells. The researchers concluded that these findings suggest blueberry intake may reduce colon cancer risk. Study 2, breast cancer. A study published by the American Association for Cancer Research examined blueberry supplementation against estrogen-mediated mammary tumor development. In the control group, tumor incidence reached 96%. In the group receiving a blueberry-supplemented diet in preventive mode, tumor incidence fell to 61% with higher blueberry doses. Tumor multiplicity. The number of tumors per animal was reduced by 51%. Tumor volume was reduced by 45% in preventive mode and 37% in therapeutic mode, meaning even after tumors had already formed. Study 3, melanoma cells. Researchers found that blueberry anthocyanin and anthocyanidin extracts inhibited the proliferation of B16, F10 melanoma cells and triggered apoptosis, programmed cell death, in those cells. Both anthocyanin and anthocyanidin extracts showed tumor inhibitory effects, with the anthocyanidin extracts showing particularly strong activity against metastatic melanoma cells. Study 4. Triple-negative breast cancer. This is one of the most aggressive and difficult-to-treat subtypes of breast cancer. Anthocyanidins from berries, including blueberries, were shown to inhibit cell cycle progression in triple-negative breast cancer cells and suppress NF-κB activation, a molecular pathway associated with cancer cell survival and resistance to treatment. Preliminary results from a colon cancer clinical trial showed 50% regression rates of rectal polyps in the treatment group. Now, let me be very clear about what these studies are and what they are not. The majority of these results come from cell culture studies and animal models. They show that blueberry compounds are biologically active against cancer cells under laboratory conditions. They do not prove that eating blueberries prevents cancer in humans. Human clinical trials on specific cancer outcomes require many years, large populations, and carefully controlled conditions that have not yet been fully achieved for blueberries. What we can say, and what is supported by the combination of mechanistic laboratory data, animal study data, and human population data, is that blueberry consumption is associated with meaningful biological effects that are directionally consistent with cancer prevention. The mechanisms are understood. The effects are reproducible across independent laboratories. And the population-level associations between anthocyanin consumption and reduced cancer risk are consistent across multiple large studies. Defense System 4, the inflammation connection. One of the most important numbers in the blueberry research relates not to cancer cells directly, but to chronic inflammation, the underlying condition that Dr. Lee identifies as one of the primary drivers of cancer, heart disease, and accelerated aging after 50. The malvidin anthocyanins in blueberries have been shown to inhibit tumor necrosis factor alpha-induced inflammatory signaling in endothelial cells with remarkable potency. At higher concentrations, MV3 glucoside inhibited more than 90% of 3 key inflammatory protein expressions simultaneously. Chronic low-grade inflammation is not something you feel acutely. It does not produce obvious symptoms, but it is working continuously in the background, weakening your immune system, damaging your blood vessels, accelerating cellular aging, and creating the biological conditions in which cancer can more easily develop and spread. Foods that reduce chronic inflammation are, in Dr. Lai's framework, foods that strengthen all 5 defense systems simultaneously, because inflammation is an underlying driver of dysfunction in all 5. This is why Dr. Lee categorizes blueberries as a Grand Slam food. Not because they are especially potent against any one cancer type in any one study, but because they work across multiple defense systems simultaneously: immunity, angiogenesis, DNA protection, and inflammation control, through well-understood biological mechanisms. Now let us talk about something that is practically relevant and often overlooked: the difference between fresh and frozen blueberries and between cultivated and wild varieties. Most people buy large, cultivated blueberries, the kind that are soft, sweet, and available fresh in the produce section year-round. These are genuinely nutritious, but from an anthocyanin perspective, they are not the optimal choice. Wild blueberries, found in the frozen section of most major grocery stores under brands like Wyman's or in store-brand frozen fruit sections, are significantly smaller, more intensely pigmented, and measurably higher in anthocyanin content than their cultivated counterparts. The deep purple-blue pigment is the anthocyanin. More pigment means more of the compound. Ounce for ounce, wild blueberries deliver more of the bioactive compounds responsible for the health effects we have been discussing. And here is the good news about frozen. Freezing does not destroy anthocyanins. In fact, the freezing and thawing process can actually break down cell walls in the berry, potentially increasing the bioavailability of anthocyanins when consumed. A 2011 review of frozen fruit nutritional content found that frozen fruits often retain comparable or superior levels of key nutrients compared to fresh produce that has been stored for several days after harvest. The practical implication: buy frozen wild blueberries. Keep a bag in your freezer. They are cheaper than fresh, available year-round regardless of season, and from an anthocyanin standpoint, they are likely the most potent option available at your grocery store. Let me now give you the food combination protocol that maximizes blueberries' effectiveness within Dr. Lai's framework. Combination 1. Blueberries with plain live culture yogurt. We discussed yogurt in the previous video. It grows Akkermansia, the longevity bacterium, and measurably improves insulin sensitivity and metabolic markers. Blueberries add anthocyanin delivery to the combination, and the fiber in blueberries acts as a prebiotic, feeding the beneficial bacteria the yogurt introduces. 3 defense systems: immunity, microbiome, angiogenesis, in one bowl. Combination 2. Blueberries with dark chocolate, 70% cacao or above. Dark chocolate doubles circulating stem cells, as we covered in the stem cell video. It also contains polyphenols with anti-angiogenic activity. Combined with blueberries, you are simultaneously hitting stem cell regeneration, natural killer cell immunity, angiogenesis defense, and DNA protection. This is what Dr. Lee means by food synergy. 4 defense systems in what is, functionally, a snack. Combination 3, blueberries with oatmeal. Oatmeal contains beta-glucans, soluble fiber that feeds beneficial gut bacteria, supports immune cell production, and has been shown to reduce LDL cholesterol. Adding wild blueberries to morning oatmeal means starting the day with microbiome support, anthocyanin delivery, and beta-glucan immune support simultaneously. 2 defense systems before you leave the house. Combination 4, blueberries with green tea. Both contain polyphenols. Green tea delivers EGCG, one of the most potent anti-angiogenic compounds in the food supply. Blueberries deliver anthocyanins working through different but complementary molecular pathways. Consuming both together is not redundant. It is additive. They are likely hitting different molecular targets within the same defense system, producing a broader and potentially stronger combined effect. Let me close with something that I think is worth saying directly. In 25 years of practicing medicine, I have watched a consistent pattern. Patients who consistently ate well, not perfectly, not according to some complicated protocol, but consistently included foods like blueberries, green tea, fermented foods, and cruciferous vegetables, showed measurably better health outcomes over the long term than patients who did not. Not dramatically better in any single year, but meaningfully better over decades. The science Dr. Lee has dedicated his career to building tells us why. It is not magic. It is biology. Anthocyanins in blueberries boost natural killer cells. They inhibit inflammatory signaling by 86 to 98% in the molecular studies. They suppress colon cancer cell growth by more than 50% in laboratory testing. They reduce tumor incidence in breast cancer models by 35% and tumor volume by 45%, consistently, across independent laboratories, using well-understood mechanisms. Does that mean eating blueberries guarantees you will not get cancer? No. Nothing does. But it means that every handful of blueberries you eat is doing real, documented, biologically meaningful work inside your body. And that consistent daily habit, accumulated over months and years, is one of the most powerful and most accessible health investments you can make. A handful of wild blueberries, every day, before your walk, in your oatmeal, with your yogurt, on top of your dark chocolate. Starting today, tomorrow on Thrive After 50, we go deep on dark chocolate. Not as a treat, but as a scientifically documented health food that activates your stem cells, supports angiogenesis defense, and feeds your gut microbiome simultaneously. We are going to look at the specific cacao percentage that matters, the difference between commercial and artisan chocolate, and why the way most people eat chocolate is eliminating most of its health benefits before it even reaches their gut. Subscribe to Thrive After 50 so you do not miss it. I will see you tomorrow. All content is for educational purposes only and is inspired by the published research of Dr. William Lee and the Angiogenesis Foundation. Thrive After 50 is an independent educational channel and is not affiliated with, endorsed by, or officially connected to Dr. William Lee in any way. Always consult your own physician for personal medical advice.